rice. After that, he moved to the Department of Biochemistry at University of Wisconsin, Madison, USA, to pursue his postdoctoral studies on molecular chaperones of budding yeast. After spending about 6 years in USA, Dr. Sahi joined the Faculty of Biological Sciences at IISER Bhopal. His research work revolves around understanding the functionality of molecular chaperones in plants and budding yeast. Besides research, Dr. Sahi is passionate about teaching undergraduate students. So welcome to Dr. Chandan Shahi. And I
Thank you.
would request to please start your talk, which is on yeast and yeast used as a model for drug screening. Please, over to Sai sir.
Thank you, Dr. Pawar. This is really a pleasure being as a panelist here. I'm sure, I mean, I'm not an expert on this, but definitely we work with
Yeah.
budding yeast. And what I have learned from you all in a last couple of years, I think that will also be obvious from my presentation today. Um, is my screen visible to all of you?
Yeah, yes, yes, sir.
Okay. Um, so as Dr. Pawar said, so first I'd like to welcome you all, and this is really a good initiative to to do keep on doing some teaching during this pandemic situation. And um, um, Dr. Pawar is a very good friend, and I have learned so much about pharmacology, pharmacy, and all. I mean, these terms was so I was not familiar with this, and now we do some of the work. And towards the end of my presentation, some of the basic things that we do, and towards the end of my presentation, I would like to show some of the results that we have published together with Dr. Pawar, and through him, I got into some more collaborations, in and around Bhopal and some of the
Thank
pharmacies
you,
we
sir.
do. Okay, so the topic of my presentation today is yeast-based molecular markers and drug screening. Actually, so this is the broad topic, and I have this topic distributed in two broad, I have two broad topics within this. And the first topic, I would like to have a more introductory section on use of budding yeast as a model, how and why yeast is a preferred model system to study these kind of molecular screening methods, what power we have with yeast that is not there with any other model organism available so far, what makes yeast today. And then toward the end, I will like to share, I mean, I'm being a researcher, I'm a little selfish that I want to share some of my own work and some of the collaborative work that we have been doing together towards the same common goal, is using yeast as a molecular use some of the markers as for using in the drug discovery and drug screening actually. So you see, this is a budding yeast colony, a small colony of budding yeast, and you see these are different, new buds, new cells are there, old cells are there, depending on the budding of this. This is a scanning electron micrograph. So before we go into the real detail of this, you see why yeast? Why we use yeast? What is the reason that yeast became so popular as a model system? Um, these are some of the reasons. This is a very simple eukaryotic model, and when I say eukaryote, the reason it is a unicellular eukaryote, and it has a very small genome. So we know completely about its genome. We know all the genes that are encoded by yeast, and the functionality of most of the genes, if not all, has been completely worked out. Um, Besides that, it's not just the single genes that we know of, the interaction between some of these genes, because you have studied, most of you would have studied in your class that genes don't work in isolation. Rather, they work as a group, sometimes in a pathway, and the genes also interact with each other. So the genetic map of these genes have also been established, well established actually in budding yeast. The sequence, the genome is completely sequenced. Actually, this was the first eukaryotic genome to be sequenced, and we know all the functionality of all the genes. So you really know what you are looking at when you have these different genotypes available in your hand. One of the things that makes yeast so important is that a lot of these genes that are present in yeast, because it's an eukaryote, a lot of these genes are conserved in higher eukaryotes, including humans. And this makes this organism very, very important, because you can ask fundamental questions about the working, the physiology, the molecular biology, the genetics, and the physiological aspects of those genes using yeast as a model. You'll be surprised to know that the first gene to be involved in cancer was identified in yeast first. So a lot of people, sometimes think why yeast is one of the things that, cell cycle genes, which have a very important role in cancer development and oncogenesis, were first identified in budding yeast model. So this gives the yeast a very, it's a very powerful model to understand these fundamental cell biology things that you cannot do in higher organisms or other cells where genetic manipulation methods are not that established, the resources are not there, and in yeast you can do it rather very easily. And then some of that I will talk in more detail later on. So you can do a lot of genetic manipulations very, very easily. In overnight, you can make mutations, you can make knockouts, overexpression constructs, and so on and so forth. So it is very easy. Now, these are very simple things, but why we yeast? Actually, you will be surprised to know that the things that I was telling earlier about it being a simple eukaryotic model and all those things came much later. I mean, people did characterized. These things came when the molecular biology and genetic characterization of yeast happened. But even before that, people have been using yeast for a lot of different purposes, right? Making wine, beer, and breads, right? This has been used for thousands of years. And the reason yeast became a model was not to study cell biology things. Actually, it was not to study human diseases. It was not to study physiological aspects of eukaryotic cells. It was used to enhance its properties as a commercial properties in terms of bread making or wine making or beer making. This is something very important. And this is how a research area grows. It had a commercial importance, and so people started studying it in more detail, and then more and more funding came, and then yeast became a model other than studying wine preparation and beer preparation also. So this is something that to be learned in as an applied aspect also. So yeast attracted a lot of funding, and yeast attracted a lot of resources in terms of people working on this and so on and so forth, and then yeast became such a good and a powerful model. No, besides knowing more about these applied aspects of yeast has been the source of most number of Nobel prizes. Actually, this is also something very, very important. You would think that this is a fundamental thing, a fundamental process, very basic organism to study fundamental processes in eukaryotes. And this is why, if you compare the number of Nobel prizes available to be given in different organisms, yeast has the most number of Nobel prizes. This is not a very updated list, but you can see that 4 to 5 Nobel prizes have been given to this eukaryote. Compare it with human research or human research, you see that it is huge. You don't find any human basic fundamental discoveries being made in more complex eukaryotes for obvious reasons, right? So before we go into the details of resources and why yeast is so useful, I want to very briefly go through the model and how this model is studied. You can see that yeast. Interestingly, yeast is a very interesting organism, because yeast can exist very happily as a haploid organism as well as a diploid organism. And this is a very important aspect of yeast, because you can study non-essential genes as well as essential genes in very efficient way using these haploid and diploid organisms. which is not possible in any other eukaryote, because in all other eukaryotes that we know of, the organism is a diploid, that model organism that we are talking about. And studying the action, the studying the function of essential genes becomes a problem, because when you knock out those genes, the organisms are dead. So this is a very powerful thing that you can do in budding yeast, right? So this is, yeast does not have sexes. It has mating types, mating type A and mating type alpha, and you can do simple mating experiments by taking two different genotypes, and you can study, you can do a crossing experiment that is done within 4 hours in yeast. So you can make a diploid organism combining two different genes or knockouts or phenotypes, and within 4 hours you get this diploid, and which diploid, you can, now this diploid can germinate to make a diploid organism. And you can keep this diploid organism as you want in the cell, on the petri plate for like several years in deep freezer, a couple of weeks on plates. So you have a very stable system. You cannot, you can freeze these cells, and you have the genotype in your hand. Whenever required, you can, just stress these cells, and these cells will now sporulate and make these tetrads, which is just after the meiosis. And once this meiosis happen, you have the desired combinations of genes segregating, and you can, do simple, this is a very simple experiment that can be done within 1 week to make a desired knockout strain, a knockdown strain, and so on and so forth, to study the function of a particular gene. So this system is very easy. um, So this is something that, as a model, is very well established. There are some problems with budding yeast, which have been, taken care of in the last several years when people started working, because this yeast has a tendency to switch the mating type. The wild type yeast that we study has a, if it is alone, if it is lonely, it will switch on the mating type, and it will become of a different mating type, right? This is present in yeast that is present in the wild. But researchers have, you cannot work, you cannot do genetics with a strain that can switch its mating type, or which can switch its sex, right? So people have genetically manipulated yeast to delete some of these fundamental genes, cell cycle genes, and recombination related genes that do not let this switching happen. And so the strains that we used in the lab, this is very important, that those strains have been genetically modified to be more stable over several years in the deep freezer and all. So you can do very reliable genetic experiments with that. So this is what I was telling, that in a population of cells, if you have a single lonely spore which is of alpha type, then very quickly it switches, undergoes a recombination switch, and it will change to a type, and now it can undergo a sexual reproduction. So this is something very remarkable with yeast, which is not present in a lot of different organisms. So it is just being lonely, it switches its sex, it changes its sex. But this does not happen in the wild type strains that are used regularly in the laboratory, because these are genetically manipulated. Now, because of some of these features that I'm just trying to tell you, that yeast has been extensively used to to study genetic circuits, right? You can mutate a gene, you can mutate a complete pathway. There people have generated quadruple knockout, triple knockouts, double knockouts, to completely remove the whole pathway and study its function in yeast. You know when you do these kind of deletion experiments in yeast, this gives you a powerful approach to, for example, testing the functionality of human genes in yeast. And there are several very good examples where they have made humanized yeast. You must have heard something like this for humanized rat models, humanized mice models, where some of the human genes have been put in the mouse. But and mouse is still a mammal, right? It is, it's not very far from humans in terms of its in the phylogenetic tree. But yeast is a very distant organism. What I'm trying to tell you, that using different knockouts in yeast and doing yeast complementation assays, people have established functionality of human genes in diseases and in specific cellular processes in using budding yeast model. So this gives you this idea of what you can do with yeast. You can have a knockout of a cell cycle gene, take a cell cycle gene from humans and put it in yeast, and you will know, okay, this is does the same thing, in it works in the same way, it has these interacting partners and so on and so forth. So you can study what that cell cycle gene is doing in humans by testing it in yeast very, very easily. People have mutated every gene, and Dr. Pawar also knows that we have the collection of all 6,000 knockout genes in our lab. And it is surprising to know that not deletion of not all the genes in yeast results into a phenotype, which means that this was something that people did not know earlier, when before working on yeast, that people thought that whatever genes have evolved for millions of years in an organism should be very, very important. But now we know that no, we don't need a lot of these genes at all the times. Maybe they are required at very specific times during the life cycle, or under very specific conditions. So these are, which means that the cells have a way of buffering. These are some fundamental ideas that came from studying budding yeast. Um, and I was saying that some of these modifiers or enhancers of human diseases, like the Huntington model, yeast has been generated, right? When you take the Huntington, which causes Huntington, which is a neurodegenerative disease disorder, you can have alpha synuclein, which causes a neurodegenerative. You can take Parkinson's, Alzheimer's. You will be surprised to know that all these genes have been put in yeast to design these kind of neurogenetic, model systems in yeast. And now you can study the functionality of all 6,000 genes in yeast, which genes result into more propensity of the disease or reduced propensity of the disease, and so on and so forth. And then once you are done with these kind of screenings in yeast, now you can graduate to the mammalian systems or cell line systems in more complex systems, to do this kind of analysis with a more targeted analysis. So yeast gives you this power of doing this kind of high throughput screens, which you can do very, very rapidly, in a very less time, in a more efficient way, by also understanding the mechanism of functionality of some of these genes. Haploinsufficiency is a very interesting thing, because now we know that a lot of these genes, which are present in humans as as a diploid copy, some of these genes, even if they are in a haploid copy, only if one of the copies are deleted or mutated, then also it results into a disease, which is surprising, and which is not something that what Mendel said, right? So yeast being a diploid, you can study the diseases, the haploinsufficient diseases in yeast more easily compared to human cells. This is something that is coming up very, very interestingly, and we are also doing some work on haploinsufficient associated diseases in using yeast as a model. Dosage imbalance, again, you will be, you are aware that, for example, if you talk about cancers, some of these cancers result into higher expression of a gene, right? For example, tumor oncogenes, right? Some of these genes, when they are upregulated, they result into cancer. There are tumor suppressor genes, which they, when they are upregulated, they suppress tumor formation, right? So you can use these kind of strategies to overexpress specific genes and see if the cells start proliferating undiscriminately. And you can study cancer using gene expression studies in budding yeast, right? So these are all the things that make yeast so very interesting. Um, so this is something that I like to show. This is a very old image, and you see that if you look at the first, um, uh, if you see this, the middle panel, you see that there are two genes here, EOS1 and IZH2. If you mutate any one of them, the cells don't care, right? They are very similar to the wild type. But when you mutate both of them, they result into a sick phenotype. What this means? This means that both the genes have a redundant function. You need to get rid of both of them to study the functionality of both these genes, right? So this is called genetic interaction studies. Try doing this kind of a study in a human cell. It would be a nightmare. Very, very tough, right? In yeast, as I said, these kind of knockouts can be made, in less than a week, in a couple of days, actually. So you can do these kind of genetic studies using yeast. And this is what people have been doing. They have been able to map the genetic interactions, whether the genetic interactions are negative with each other, whether the pathway is essential or not. whether the pathway has a parallel pathway operating in the cell. You can do different set of knockout studies in different combinations. And this people have done using robotics in some of the labs in the world, and there are some of similar studies are being done in India also using robotics, that you can combine mutations in all possible combinations and study what happens to them. The reason is, why I'm talking about this. This is something which is more important. Probably none of you work with yeast. So why would you care? The reason you should care is that, must have heard that, a mutation can have effect. People get a mutation, but some of those mutations do not result into a disease in a particular individual, but the same mutation results into lethality in a different individual, right? How this happens? The reason it happens that because even though one gene has a very important role, it only manifests its phenotype when a different set of genetic mutations are there in that organism. For example, a gene X mutated will cause cancer only when a gene Y is also mutated. How do you know this? How do you know that this gene X mutation will result into cancer in which are different combinations? It is not possible to do this kind of a study in humans, that easily. People can do this, right? People are doing, CRISPR screens, but in yeast it is very easy because people have done it over a week. With robotics, you can do this kind of analysis in a week. And people have found out that there are specific genes that either increase the disease formation or decrease the disease formation in a particular genetic setup. And this is how people have generated genetic maps, right? So this is what I was trying to tell you, right? What happens in different combinations. You don't need to know the details of this work, but what I'm trying to tell you is that a gene can have a phenotype in a combination when it is in a particular combination with other genes, right? And this is a very important idea for a disease, right? Like cancer, which there is no fixed rule with cancer, right? I know a lot of people who did everything in their life, smoked and everything, and then nothing happened because their genetic makeup was such that they were able to tolerate that. And there are a lot of people who are perfectly, they just they just die, right? Nothing, a very young age they die, no age is not there, and the person was completely not indulging into any of these activities. What this tells us? This tells us that it is something in our genome, this is something in our genetic setup that makes us vulnerable or resistant to a particular disease, right? So this, how do we do this? In this time of this COVID pandemic also, we know that one person can die with COVID, but the other person does not even know that he or she has got the infection, because our genetic makeup is different, and that results into different immunities. So if we know what is happening in this case, you will be surprised to know that people are doing these kind of genomic studies using COVID. If you look at some of the recent publications that are coming up in the good journals, what is inside our genetic makeup, right? So this is something that people have done very, very extensively using yeast as a model. What makes yeast so useful? Because in yeast there are different set of resources, right? And some of these resources are that you can do yeast interaction studies. You can see which proteins interact with which protein. We have the whole collection, the deletion mutants are there. We have repressible constructs are there. We can do transposon mutation very easily. We know at the proteome level which proteins are localized in which part of the cell. This also gives us a lot of information about the functionality of a protein. For example, if a protein is in the nucleus, it will have more a gene regulation function or a chromosomal stability or chromosomal segregation. And if a protein is in the mitochondria, it will have a more mitochondrial function or something like that. So people have tagged all the genes in yeast and done the comprehensive protein expression and protein localization studies. You can have tag constructs of yeast genes, and you can do pull downs, right? What proteins interact with a particular protein, right? A lot of times, for example, if I'm indulging into a bad habit, I will have interaction with bad people. If I'm indulging into good habits, I will have interactions with more academicians or something like a good interactor, right? So the way you interact tells a lot about you. In the same way, the interaction map of the proteins tells a lot about the functionality of that protein, right? And this has been done for all the yeast 6,000 genes in yeast, because tap tag pull downs and mass spectrometry has been done for all the genes in yeast. So this is something has generated a lot of resource, which is not only applicable for understanding basic and simple pathways, but also has a higher level implications in humans, disease, a lot of these things. So, you can do, as I was telling you earlier, that you can not only study the deletion phenotype of a single gene, but you can also do synthetic genetic interaction, right? As I was telling, what happens when X is mutated with Y, and what happens when X is mutated with A? How these do genes interact with each other, right? So people have mutated all these synthetic. So this is called a synthetic genetic array, where different combinations of genes can be made, and you can study this, right? And this is mostly done at the robotics. Robots do this. You just have to program, and you take the cells and give it to them, and they will make all these knockouts within a week, right? And this kind of service is also available. The reason I'm always emphasizing on synthetic genetic arrays is because a lot of times single genes do not tell you anything, right? A lot of times the genes need to be seen as an interactor with other genes, right? For example, I can interact with my family in a different way, and I can interact with my students in a different way, and I can interact with with office staff in a different way, right? So how a gene interacts with different genes, why, what is the outcome? If I'm interacting with my family, I'm more into a family business. If I'm interacting with my students, I'm talking about research and I'm talking about papers. If I'm interacting with a person at the traffic light, I'm probably fighting with someone, right? So my interactions tells a lot about my functionality, and this is very, very important. So genetic interactions are very, very powerful. So this is how it looks, right? This is how the robot does it, right? The robot marks these cells by, so these are different genotypes on different genetic makeups. So as you can, I don't know whether you can see my cursor, you see that in this here the cells are not there, so the cells is dead. It means that these two genetic genes, when you combine them together, the two gene knockouts, the cells die. I don't know whether you can see my cursor or not. So
Yes, sir.
likewise,
Yes, seen, sir.
great.
Yeah.
And likewise, if you see a different set of cells, you see that these two cells are very small, right? Which means that, so these are all in duplicates, which means that these genetic combination results into a sick phenotype. And why this sick phenotype is there? Now you can do this analysis. Imagine doing this kind of, so this plate has several thousand colonies, several thousand genetic combinations on one plate, 96 well plate. So not a 96, this is a microtiter plate. What I'm trying to emphasize is that on one microtiter plate you can analyze several thousand genetic combinations, and you can select, okay, I want this one, I want this one, I want this one, I want this one. And then you can study in more detailed analysis of that screen. So yeast has been used as a screen, right? This is called a synthetic genetic screen, or a SGA screen, to look for phenotypes, right? So very fast, it becomes very, very fast. You can know, how do you analyze this? Once you get these colonies, you can check for you can do this at the genome level also. You can just pool the cells, and you can look for specific markers for their expression, and tell which cells are growing slowly, which cells are growing faster, because all these yeast cells are barcoded with a very unique nucleotide sequence. So you can look at their growth status and all those things by using simple PCR methods. By using simple quantitative PCR, you can tell what happens when this genetic combination comes together. So this gives you a very powerful method of analyzing several thousand genetic combinations in one go, right? This is the methodology of the SGA screen that is being used. And we also use this kind of a screen in the lab, but we don't have the robot, so we do this manually, but this can be done by using robotics also. What this gives us at the end, this is what I was telling, that this gene K interacts in different ways with with three other genes, right? For example, it can interact with genes within the family or within the pathway. It can interact with genes in different ways in terms of positive protein interaction. genetic interaction, positive interaction, negative interactions. I might be interacting very positively with my students. I might be encouraging them, but when I go home, I might be fighting with my family, right? This tells a lot about me. This tells a lot about the gene, right? How this works, what is the working mechanism with different set of genes with different pathways, and this gives a lot of information. Using this kind of phenotyping along with microscopy as well as a lot of computational algorithms, you can come up with these kind of maps which are called genetic interaction maps for separate genes. So you see here there are a couple of genes, maybe a dozen of genes, are a map of a couple of genes has been shown. Now, the moment you do this at the genomic level at the yeast, you can get more saturated maps, right? This kind of information is not available, is not possible for any other organism other than yeast. And now people are coming up with 1 billion genetic interactions, and which is something that Mendel would have never imagined, how individual genes. And Mendel just talked about capital T and a small t. Now these people are talking about high level genetic interaction makeups, very dense genetic interaction makeups, how one gene interacts with different pathways. So you see that the genes interacting in a pathway cluster together, but they also interact with other genes. This is very valuable in the sense that a gene involved in DNA repair might be involved in mitochondrial import in some way, which was previously completely unknown. So you have you can do a lot of these kind of things with, if you have this kind of resources with you with yeast. So networks have been generated, genetic maps have been generated, right? This is just the enlargement of the same image from a cover paper in Science, and this talks about the genetic interactions within the. This is the mitochondrial proteins interacting with each other. So these obviously they are closer to each other, right? For example, protein folding, cell wall biotin, they come together, but it's not that they are in family all the time. They are interacting with only each other. You see that these are networks. These are interacting networks. Some of these genes in the protein folding pathway might be interacting with some mitochondrial genes also. So this is what people have come up with. This is a remarkable achievement, I think, in last 2 decades, that have come up using yeast as a model system, using yeast as a screening system. So what I'm now gradually coming to is that, I was talking about genetic interactions, but people have also put in drugs. You can put in drugs. For example, if you put in a drug that challenges cell cycle pathways, right? If you put in drugs that challenges cell cycle pathway and you see which genes get affected more strongly using a particular drug, and we know about the functionality of that drug. For example, if we take a drug that affects N-glycosylations in the ER or the endoplasmic reticulum, then the genes which will have a function, which will have some association with the endoplasmic reticulum, will show a phenotype. Other genes might not show a phenotype, right? So this way, what people did was they did a comprehensive genetic screen for all the knockouts that I was talking about earlier, and people have done very, very comprehensive screen for a variety of drugs that sorry, the drugs that target mitochondrial biogenesis, drugs that target the cytoskeleton, the drugs that target nuclear import. So this, what they have done is they have now gone to more drug oriented screen. So for example, if you pass on a drug, right, there's a approval for a drug that cures cancer or drugs that kill a particular pathogen in our body, but we don't know how it affects the different cellular pathways, right? How will you do this? You can do a screen, and high throughput screen, and then you can know that, okay, this affects, although this is working in this pathway, it is also affecting this pathway. And these kind of screens have been done in yeast very, very efficiently. Several different types of screens have been done, and not only with single knockouts, with double knockouts and quadruple knockouts also. So this is called a chemogenomic screen, and people have used this extensively to study several genes associated with a particular pathway, genes associated with a particular process for which a particular drug is available. So you can know more about the functionality of genes and pathways, and as well as about the drug. Okay, so this is called a chemogenomic screen, and for which yeast has been used extensively. So this is how it works. You provide a drug and you look for a phenotype. Your phenotype could be in terms of growth. Phenotype could be in terms of a phenotype in terms of morphology of the colonies. It could be anything, right? It depends on what is the output. But that way you can group genes, group pathways that are affected by this drug or which are challenged. If you provide the drug, these genes are very important to keep the cells functioning. So these kind of information you can go get very very fast using budding yeast model. So this is my lab at ISET Bhopal, and I want to change gears. How much time I have? I have, yeah. Okay. So at my group at ISET Bhopal, we work with a variety of different organisms, and budding yeast is one of them. And basically, my group works on protein folding and aggregation and so on and so forth. So molecular chaperones. I will skip this part very fast because I think this is very textbook thing. So protein folding is very important, and and now it is known that if protein folding is not as simple as what was proposed by and Finson, Professor M. Finson, which got him the Nobel Prize, but it also depends a lot of other factors, including mutations, diseases, and stress, and all sort of things. Protein folding is not as simple as that. I know this is not a very choreographed folding process that happens in the cells. Protein folding is more like a chaotic process that happens in a wave pool in a water park, right? So this is proteins are colliding with each other, and proteins are affecting each other in bad ways. So protein folding is a very important process, and it requires molecular chaperones, and this is where we come and we work with molecular chaperones. So what are molecular chaperones? Molecular chaperones are proteins that help other proteins. So this is, I think, which you need to know a little bit, right? These are proteins that help other proteins. How do they help? They help in maintaining their behavior as shown in this slide. This behavior, maintaining this behavior, this term comes from from the place where these proteins are born, right, in the cradle, to the place that these proteins die. So the protein chaperones are helping other proteins from the cradle to their grave, right? So they are important at every step of protein folding and transport, stability, activity, till their degradation. So protein chaperones are very, very important. And now we know that chaperones are not only important for protein folding, but they are also important for transport, preventing aggregation, solubilizing proteins in an aggregate, refolding them, and so on and so forth. So they have a very broad effect. If you compromise some of these effects, you can result into risk, right, of disease, risk of protein proteostasis collapse, aging, and it is related to all sort of things in our bodies, right? So that's why understanding protein chaperones is important. No, we know that these chaperones are of different families, and we know, and there are these chaperones belong to different families. They have been categorized depending on their molecular weight. And what we are trying to do, as I was telling you earlier, that these chaperones do not work alone. They work as a collaborative unit, right? They collaborate with each other. They make a network. As I was telling you earlier in the previous slide, that these genes interact with each other. Proteins also work together, especially chaperones. They help each other as a community. They work inside the cell. They work together. Yeast has a lot of different chaperones, right? And they are involved in different cellular pathways and all. And there's a lot of information already available in the literature about the functionality of these proteins in yeast, right? Some of these genes are essential, some of these are non-essential, and some of these genes, if you knock it out, they don't have no effect in the cell. They have no effect on the cell. The cells just don't care whether the chaperone is there or not. So what people did some time back, as I was telling you earlier, they wanted to see whether some of these proteins have a defect under a particular condition, especially under particular drugs, particular inhibitors. And what they found was, indeed, some of these chaperones which otherwise do not have a phenotype, if you knock them out, are compromised. They result into compromised growth in the cell when you knock them out. And you grow them in the presence of a particular drug. Now people had more knowledge about some of these functions in yeast, about their functionality in some pathways in yeast, right? So, I think I will rush through it, and I don't want to talk too much about this work, because this work has been partially published, and this work was I started long time back, where what we did was we took several drugs. We did a chemogenomic screen for chaperones. We took several drugs we purchased it commercially, and we screened all the yeast cells, the chaperone cells, and we came up with several new ideas which was not previously known, that some of these chaperones may have a role in some of these specific pathways which are perturbed by these drugs, and we are pursuing it more aggressively in the lab these days. So I will rush through it. I don't want to talk in too much detail. um, uh, Specifically, I want to talk about this work which has been recently published. So this is a drug which is causes immunosuppression. So this drug causes immunosuppression. This is called FTY-720. And the way it causes immunosuppression is that it stabilizes membrane proteins at the plasma membrane. This is the information that we knew. And several years back, around 17 years back, someone showed that if you overexpress one of these HSP40s called CAJ1, which we also work with, it results into a reduced tolerance to this drug. And this drug has a very important role in immunosuppression, and this is given to in some way to transplant patients and all, right? So we were so intrigued about the fact that why overexpression of this chaperone results into more tolerance against this drug. So what we found, I'm not going to talk in too much detail, but I'll tell you what we found. This work has been recently published. What we found that when we overexpress this HSP chaperone, it results into a stabilization of membrane proteins. And this membrane protein could be a ABC drug, ABC transporter at the plasma membrane, which is pumping out the drugs, right? So what the idea is that when we overexpress these HSP chaperones, we believe that some of these membrane proteins could be stabilized in some way, and that results into reduced sensitivity of the cells to this immunosuppressive drug. Now, why I'm talking about this? Because using yeast, you can talk about the mechanism and physiology of this gene, which you cannot do using human cells that easily. So we have done complete work on this gene, and this work has been published. And we now know that this protein associates with the plasma membrane. So this is a membrane associated experiment, and we were finally able to get this work out last week, as well as a work on in the last year. So some of this work was done in collaboration with my friend Fumiyoshi Abe in Tokyo, University of Tokyo, and very interesting study that started with a chemogenomic screen. This is what I'm trying to tell. Now, why I'm here? I'm not here to talk about the chaperones. I'm not here to talk about much about this. Yes, plants are packed with secondary metabolites. The term secondary metabolite is itself very debatable, because it was coined to say that these secondary metabolites are not essential. They do not have any important role. They are not essential for the plant. But now we know that secondary metabolites are very, very important, and sometimes essential, because some of these secondary metabolites are the pathways into which chlorophyll pigment is linked, different flavonoids are linked, different types of secondary metabolites are produced by the plants. And this is a very, very rich resource, right? We talk about buying chemicals from Sigma and studying gene function, but plants provide a lot of this different resource for free, right? And also it has a lot of medical applications, which and this is where I got in touch with Dr. Rajesh Pawar. And there's a lot of work here. I'm not the right person to talk about this, but this requires a very comprehensive and interdisciplinary, which is also the title of today's presentation, right? This bring people from different areas of research. For example, I am a cell biologist. People could be from pharmacology, cell biologist, computational modeling, right? Chemical synth, chemists, right? Organic synthesis, right? All these things can be done to study the functionality and activity of some of these chemical compounds present in plants. So I'd had this chance and very good luck to talk to these people, Dr. Pawar, who is right now at Truba, and Dr. Vipin Dote, who is currently at VNS. And I just want to tell you, I want to acknowledge these guys for my understanding of this and my appreciation for these kind of drugs and compounds that are made in plants. And this is the work that was done in collaboration with Dr. Pawar, and with his permission, I would like to briefly talk about some of the work which was done in my lab, where we showed that some of these extracts prepared from Sida cordifolia were almost as efficient as ascorbic acid as a in terms of quenching superoxide radicals in. So it was very easy for us to do. I know the hard part was done by them, but using yeast, you can look at these molecular events that can happen in the cells. So at the molecular level, at the cellular level, we were able to unequivocally show that these drugs have the same activity, or these extracts have the same activity as ascorbic acid. Probably in some case we found that it was slightly better also, which is we cannot imagine what is the implication of this thing, right? Bringing together yeast as a screen for these kind of studies has a lot of implications. This was published in 2019, and last year we published another paper with Dr. Vipin Dhote, where we showed that another extract, noscapine, alleviates cerebral damage in ischemia repercussion injury in rats. The idea here was again using yeast, and once this was shown, they did some work with rats and show that this compound was indeed very, very effective. So with this, I would like to really thank Dr. Pawar for giving me a chance to talk about my work, as well as tell you a little bit about yeast in general. This image is a little old, but I wanted to acknowledge my undergrads who are the driving force in my lab, and I love interacting with them because they are free of any inhibitions. They think independently. So this is my group. There are some PhD students, and this part of my lab are the undergrads who contribute significantly to a lot of yeast work being done in the lab. A lot of time these screens. My lab is decently funded with from SCRB and Department of Biotechnology, and we also enjoy some intramural funds from ISER Bhopal. I am not sure whether I will be, I have time to answer some of the questions if there might be. Otherwise, I would, thank you all for your time, and thank you all for being here on this platform. Thank you very much.
Thank you, sir. Thank you for your nice talk about the yeast as a model to minimize the animal burden when we are going to screen any molecule on the animals. So yeast could be a very good model to drug screen, for drug screen.
Yes.
So just I would like to know, can it be possible to screen out neurodegenerative disorders using yeast as a model?
Exactly. So as I told during my talk, that there are some neurodegenerative models also available in yeast. So what people have been trying to do is people have used these kind of aggregation models, neurodegenerative models. They have put it in yeast, and as some of the examples I said was Huntington and synuclein, alpha synuclein. What they have done is they have put these aggregates in yeast and looked for different genes combinations, single deletion, the whole genome, and see which loss of which genes results into more aggregation and more disease pathogenesis, right?
Okay.
Using yeast. So now what can be done is, you put some of these extracts from plants that you guys are making, and that will be really interesting to learn if, we have already identified some proteins in plants that make these kind of aggregates. So and what I'll not be surprised if I know that some of these extracts that are present in plants may be inhibiting some of these aggregations in yeast. So as a screen, in combination with these extracts as well as genetic combinations, these screens can be done. Yes, for sure. We can combine these kind of things.
Thank you. The overall correlation between these activities is oxidative stress, I think.
For the work that I presented.
Yeah,
for that it is oxidative stress, and we have used some genes which are very sensitive to oxidative stress. So we have shown that if you provide these extracts, the cells can tolerate, even those cells can tolerate the oxidative stress much better than no extracts without the extracts.
Okay. Thank you for your nice talk
Thank you so much.
on yeast as a model. So thank you, sir. We will meet again with another topic
Sure.
in future.
Sure.
Thank you for
so much.
giving us time. Okay, thank you.
Okay. So I can leave now?
Okay, thank you.